So can you chat and say yes or no in the chat, please? Make sure that we all can see it. Yes.
Yo no la veo. Okay.
¿Quién no la ve?
¿La presentación o ahora?
Some do, others don't, somehow.
Yo tampoco puedo ver la presentación.
Yo tampoco.
Okay. So, okay, so we have some yes and some no.
Excuse me, Dr. Maldonado, maybe all the participants can put the presentation in pin. You can do with pin the presentation individual. Maybe it's an option for you.
Okay. Well, not necessarily. Well, Dr. Kinsler, could you come out again and try again so we do the same procedure, please?
Okay. So how far back do you want me to go? I'm now, do you want me to actually go back to, can you see me?
Not share.
I can see you.
Huh?
I can see you. Can you see me?
I don't see you. Do you see me?
I see you, yes.
Okay. So I see you now.
So you want me to stop sharing? Because that would be the only.
Stop sharing, please.
Okay.
And then we will do the whole process once again.
Now can you see me?
Yes, I do.
Okay, good. Okay.
So
So
we go back to the
you want me to hit present now?
right. Yeah.
You see.
And then go to one window, right?
Oh wait. I'm looking for the the present now button actually appears and then it disappears. Okay, present now.
Yes.
A window, right? Not my entire screen. Okay.
And go. And
Can
then,
you
yes.
see that?
It's coming.
Now it works.
Mm-hmm. Great.
Now
Sí, se ve Maldonado.
Now everybody see. That's fine. That's fine then.
now you can see it?
Yes, sir.
Okay.
Okay.
Okay, so I think we're ready because it's already 4 minutes after 5. And I just wanted, first of all, to welcome you to our first seminar of our summer virtual seminar series from BP Endure. And this is a collaborative effort from different programs from the BP Endure community, including, of course, the Sony Downstate Medical Center Department, where our first guest speaker is from, and also Michigan State University, LSU Health in New Orleans, the University of Colorado, Denver, and the University of Washington in St. Louis. And therefore, for all of us, it's a perfect opportunity to come together and share new data and new science that is happening across our community. And so I would like to introduce you to our first speaker, Dr. Adam Ginsler. And Dr. Ginsler is professor of biochemistry, neurobiology of pain and addiction research in the Department of Obstetrics and Gynecology at SUNY Downstate Medical Center. And he, on behalf of SUNY Downstate, successfully spearheaded and directed the implementation of the Women's Health Research Training Grant, the first such grant in New York State. Dr. Ginsler runs a multidisciplinary research laboratory that has been funded by NIH for more than 30 years. And his approach to research emanates from the firm belief that inductive approach of clinician researchers and the deductive are complementary, not anti-aesthetically or exclusionary. Dr. Ginsler was the first to demonstrate that naturally occurring presence of morphine in neuronal tissue. And as an assistant professor in Columbia University, he demonstrated that physiological hesitation is associated in the spinal opioid antinociception and subsequently elucidated the endocrine and opioid components underlying an antinociception. So his research includes the molecular interface between ovarian sex steroids and analgesic pain mechanisms, focusing on the molecular underpinning of male-female dichotomy in the prevalence of chronic pain syndromes. So Dr. Ginsler received his doctorate degree from New York University School of Medicine and his postdoctoral work at Roche Institute. So without further ado, today, Dr. Ginsler will talk about activating endogenous opiates for pain relief, wishful thinking or reality. And please welcome Dr. Ginsler, and our all questions will be pleased to include them in the chat, and we'll have at the end a Q&A session. So Dr. Ginsler, thank you.
Well, thank you very much. I very much appreciate this invitation. I think this is a wonderful thing to happen over the summer, particularly given what we're all going through with the COVID-19 pandemic. I think that it's really rather humbling to realize that it took something like the COVID-19 pandemic to really move the opioid pandemic off the front pages. But it's still very, very much a problem. And unfortunately, when the COVID-19 pandemic winds down, as it seems to be, and hopefully it will, unfortunately, the opioid pandemic will once again be center stage. And I think part of the difficulty in dealing with the opioid pandemic is that on the one hand, the crisis was awful. No doubt that narcotics, opioids are very addictive, and many, many people die from misuse and overdose. But what makes it difficult is the fact that these are also very, very effective drugs in managing pain. And it's very, very difficult to really kind of find the right equilibrium between minimizing the horrible effects of abusing opioids and also not compromising pain management. And I think what I want to, and I emphasize this point because it's given the horrible effects of opioids and the all too many deaths that have occurred from its misuse, there is almost a backlash, and there are many clinicians that are under-managed pain because they're very, very fearful of prescribing opioids. And so I want to start by underscoring that minimizing the use of opioids, and as a consequence of that, albeit an unintended consequence, under-managing pain is itself very, very problematic. And that pain is bad, pain is weakening. And I want to just kind of go through brief examples. So undertreated pain or under-managing pain often results in poor sleep, depression, and anxiety. It alters not only emotionally, but alters actual brain structure and function. For example, it decreases gray matter in pain-transmitting areas of the brain. It alters functional connectivity of cortical regions. It impairs emotional decision-making. It decreases prefrontal and thalamic gray matter density. It decreases motivation via increased long-term depression in certain particular areas of the brain in the nucleus accumbens. And so basically, the real problem is how do you reconcile, how do you balance the medical ethical imperative to manage chronic pain with the opioid crisis? Now, in addition to pharmaceutical opioids, and in addition to sources of prescription opioids like the big drug houses like CVS and Rite Aid, etc., the brain is itself an opioid factory. The brain makes opioids. And so the first question that you have to ask is, are endogenous opioids analgesic? Yes, you would think they are, but what really is the evidence that endogenously in the body, they can actually work as opioid analgesics? So back in 1980, my laboratory demonstrated that pain thresholds are actually elevated during actual physiological pregnancy. Now, just because you get an increase in pain threshold, you can see that it progressively increases, peaking just before parturition, and then there's an abrupt decline. In non-pregnant animals that were treated in parallel, you can see that there's no change in pain threshold. Now, this, in effect, doesn't necessarily tell you that this is endogenous opioid-mediated. But what does tell you that this is exclusively opioid-mediated is the fact that this increase can be completely eliminated by the drug Narcan, which is the brand name, or the generic name is naloxone. This is a pure opioid receptor antagonist. So if an emergency medical worker comes across somebody who has been overdosed with a narcotic, they will always carry a syringe containing naloxone or Narcan. And it's truly remarkable that you can inject somebody who is overdosed and has stopped breathing. Literally within seconds, their eyes will open and they'll resume normal respiration. So this increase that you get during normal gestation, this analgesic effect, is basically completely mediated by the endogenous opioid system. Now, I will say that in this series of experiments, there are a number of reflexive pain measures. The one that we used initially back in 1980 was called flinch jump. You put an animal in a cage, and the floor of the cage is comprised of grid bars, and you apply a very, very low electric current, and you basically quantify where is the threshold current that is necessary to make the animal jump. Now, subsequent to our discovery of this effect in laboratory animals, there was several years later, there was a beautiful paper by Kogan and Spinato who applied this finding to pregnant women, and they looked at during the third trimester, and you can see this a progressive increase in the ability of these women to tolerate pressure applied to the non-dominant forearm, and that pressure was applied with a blood pressure cuff. Again, in non-pregnant animals, you could see, non-pregnant women that were tested in parallel, there's really no change in their tolerance of pressure applied to the arm. So this certainly established that you don't have to do drastic things to an animal. On the normal physiology, there are certain events which the endogenous opioid system can be activated and produce an analgesic effect, less sensitivity to pain. Now, this effect of the endogenous opioid is pretty extensive because it's effective against somatic pain, which includes electric foot shock and thermal stimuli applied to the tail. It's also effective for visceral pain, and the one that's most directly studied in the laboratory is colorectal distension. And as I just mentioned, it is also in women with pressure applied to the forearm or thermal stimuli applied to dermatomes, specific dermatomes. Now, one of the early things that we did, and this has relevance to what I'm going to be discussing with you today, is that one of the key elements in this pregnancy-induced analgesia is changes in the circulating levels of estrogen and progesterone. Because if you take non-pregnant rats and you ovariectomize them so that you eliminate the major source of estrogen and progesterone, you remove the ovaries, and then you put back estrogen and progesterone, mimicking the actual profile of change that occurs during actual physiological gestation, you actually get pretty much the same profile of analgesia, or another term for analgesia is anti-nociception. Nociception being the process of perceiving pain. And what the narcotics do is they're anti that, they block that perception. Now, this is more detail than I really was intending to show in this seminar, but I'm going to show it because I want to emphasize a basic fundamental point with respect to controlling the activity of endogenous opioids. And so, as I mentioned, a key player is circulating estrogens and progesterone. And what we found out is this circulating estrogen and progesterone can either act on the uterus, and then the major afferent nerve from the uterus is the hypogastric nerve. It transmits signals all the way up to the spinal cord, to the brain, and there are specific cell groups in the brain which then activate descending pathways, and you activate a particular kind of receptor, the alpha-2 receptor. And this actually produces analgesia. But in addition to this, the estrogens can act directly on the spinal cord, where they increase one of the endogenous opioids. In this case, it's dynorphin. And then you activate a synergistic interaction between two of the three types of opiate receptor. There are three main types of opiate receptor. They have Greek symbols mu, delta, and kappa. And the two that are activated here are relatively minor components of the three opioid receptors. And you activate a synergy between kappa and delta and the alpha-2. Now, the only reason I really put this slide up is to communicate very early that the endogenous opioid system is very carefully controlled, and that there are many, many elements and dimensions to activating it. And that really bodes well for developing drugs that might be able to tap into the system, because theoretically there are many sites at which these drugs can act. And what I'm going to do in the balance of my seminar is to highlight several specific points where we can actually intervene and therefore activate the endogenous opioids. So in addition to the example that I just showed you moments ago, where physiological gestation, and by simulating the pregnancy blood profile of estrogen and progesterone in the absence of a growing fetus, there's a lot of other evidence that suggests that endogenous opioids can produce analgesia. And I'm just going to go through some of them. So I'm sure that you're all familiar with the placebo effect. That is, a person thinks they're getting medicine, but they're really not. They may be just getting a sugar pill. But basically, the thought that they're getting medicine, in many cases, is enough to actually produce the effect of the medicine itself. And that's what's called the placebo effect. And way back in the late 1970s, it was shown that if you block the opiate receptor by Narcan or naloxone, you can actually eliminate placebo-induced post-operative dental pain. Opiate receptor block also worsens clinical pain. Placebo analgesia actually correlates with enhanced endogenous opioid activity in very particular discrete areas of the brain. With very sophisticated imaging techniques, it was shown that merely the expectation of pain relief can actually activate the mu opioid receptor. And I will tell you that the mu opioid receptor is the major opioid receptor that is targeted medicinally. So when you take drugs like morphine, or you take drugs like fentanyl or sufentanil, the analgesia that result is because these drugs are activating the mu opioid receptor. And not only will the expectation of pain relief activate an endogenous opioid system, and I find this actually incredible, is that spatially directed expectation of pain relief produces analgesia only on the body part targeted by the expectation. So for example, if you actually think that you're taking a medicine that will only work, let's say, on your right arm, then the analgesia, the placebo analgesia, will be spatially directed to that body part. So really what this establishes is proof of concept. If you take all of these observations and you combine it with the early work that my lab performed with physiological pregnancy and hormonal simulation of pregnancy, it really establishes proof of the concept that harnessing endogenous opioids can be an effective strategy for managing pain. But the real question that whoops, obviously I hit a button that I shouldn't have.
We see it. Everything's fine. What's up?
I'm sorry?
I see the presentation.
Oh, okay, I can't see the presentation. I can't see the presentation.
Okay. Did
Click on Google.
You want me to go back to click? Well, here it is. You see it now? I can see it now.
Okay.
Okay, so this establishes proof of concept that the endogenous opioids can produce analgesia. The real question becomes, is this clinically useful? Are there pharmacological strategies that one could use to actually produce an endogenous opioid analgesia in the clinic? That becomes the question. So this is our approach. What we decided to do is we asked the question, can we identify naturally occurring physiological states in which responsiveness to an injected opioid is turned on or turned off? That was the first question. And I'll give you a little preview, is that we were able to identify that, and that happens to be stages of the estrous cycle, which is analogous to the menstrual cycle in women. The second thing that we said was, can we determine the mechanism that's responsible? So if you can define a physiological state where a certain opioid no longer produces analgesia, what's the underlying mechanism that's responsible? Then we needed to validate that determinants of responsiveness to exogenous opioids, opioids injected, likely is relevant to the endogenous opioids. Okay, and then we had to make a decision. Do we use males or females? And basically, what endogenous opioid are we going to be used to studying? So what we decided, and I'll explain why, is that we decided to use females. And we also, of the endogenous opioids, the one that we decided to work with was endomorphin 2, sometimes abbreviated as EM2. This is not only the smallest endogenous opioid, being only 4 amino acids, but it's the endogenous opioid that is the most selective for the mu-opioid receptor. And this is why we chose females. We chose females because in male, basically, and in all the experiments that I'll be showing you, we actually inject the various drugs directly into the spinal cord. I'll have a schematic diagram 2 slides from here, and it'll become very clear. But we're not injecting drugs, no matter what drug we want to study, systemically, but we're injecting it right into the space between the spinal cord and the vertebrae. That space is referred to as the intrathecal space. And what you can see is that in males, which is the black bar, the EM2 is very, very analgesic. Very, very analgesic, not surprising. If we look, there are 2 main estrous cycle stages that we chose to study. The one that is the most male-like, is thought to be the most male-like, is diestrus, and the one that's characterized by higher levels of estrogen is proestrus. And this is where we began to find things that we didn't expect. So in the male, it was not surprising that spinally applied EM2 was analgesic. In a proestrus rat, which is the most female-like, the EM2 was actually equally potent. But what was really strange is basically in a diestrus animal, the EM2 had no response. And what you see here on the right just shows that it could be that the time that we were waiting after application wasn't long enough. Maybe there was just a delayed response, but not true, because if we go out even 25 minutes later, there really is still no significant analgesic response. And so that told us that if we could figure out what is the on-off mechanism, it's off, the response is off in diestrus, but it's on in proestrus. If we could figure out what that mechanism is for modulating EM2 analgesia, it could indicate to us ways to manipulate endogenous EM2 analgesia. Because if we could turn it off and on here, that might be a way to turn it off and on, not in the injected EM2, but in the EM2 that the spinal cord actually produces. The other reason we chose females is because it's widely known, widely accepted, that women have a much greater severity and incidence of chronic pain than do men. And so I highlighted a few, like migrating is 2 to 3 times more prevalent and severe in women than men, or something called fibromyalgia, which is a very generalized hypersensitivity to certain stimuli. It basically is 6 times more prevalent in women than men. And so we reasoned that maybe the reason women have more severe and more frequent episodes of chronic pain is because maybe there's a dysfunctional endogenous opioid system, and the analgesic effects of turning on endogenous opioids might be more apparent. So this now is the diagram that shows what we did. There is a space between the vertebra and the actual spinal cord. That's called the intrathecal space. And we basically go through the cisterna magna, which is the space in the lower back of the head. And we really kind of put in a very, very small cannula that goes down into the lumbar spinal cord. And in all the studies that I'll be showing you, well, not all, but most of them until the very end, we use a technique called the tail flick test. And that is, we use rats, not mice. The rat is put on a platform. The tail is positioned so that when you can shine a beam of light, and the rat keeps its tail there until it says, wow, this is now starting to become too hot. I don't like it anymore. And it moves its tail. And that's where the name flick comes from. And so what we're really measuring is a threshold response. So the question that we asked is basically, what is it about diestrus that prevents spinal EM2 from producing analgesia? Again, we want to determine what regulates the response to exogenous EM2. And once we know that, we can then form theories about what might be going on endogenously. So I'll give you a little preview. First, I'll tell you what I'm going to tell you, and then I'll actually try to convince you of it. So the absence of intrathecal EM2 analgesia during diestrus results from an active suppression. There's an actual active mechanism that clamps the response. This suppression, this active clamping, results from the activity of 3 components: estrogens, estrogen receptors, and a metabotropic glutamate receptor. And that if we could eliminate this suppression, as occurs naturally during proestrus, if you remember, diestrus had no response to EM2, but proestrus had a response equal to the male. Well, if we eliminate this suppression, then we will not only unmask EM2 analgesia, intrathecal EM2 analgesia, the EM2 that we inject in the spinal cord, but it will also serve to activate endogenous opioid analgesia. And for this, we do that study in a chronic pain model, and I'll explain momentarily why and where that comes from. So the first thing we did is that if you get an effect in proestrus, but not in diestrus, the first thing you probably think of is that it must be due to estrogen, because estrogen is higher, circulating estrogens are higher in a female during proestrus than diestrus. And to do this, what we did is that the estrogen receptors are very, very well characterized pharmacologically. And there are specific drugs that will basically block either estrogen receptor beta, and that's what's blocked by this compound called PMT, or estrogen receptor alpha, which is what's blocked by MPP. There's a third one, G15, that's a third type of receptor. To make a long story short, if you look at this, you can't distinguish between the various lines. And what that tells you is that even though in proestrus, circulating estrogens are the highest they'll be across the estrous cycle, this doesn't play a role. It has no effect. However, if you do the same experiment in diestrus, what you see is that during diestrus, you actually unmask EM2 responsiveness. So basically, if you just give the vehicle, okay, or you just give the EM2 by itself, it's not producing any antinociception. But if you inject this estrogen receptor alpha blocker, so you block one of the types of estrogen receptor, now all of a sudden EM2 becomes active. Now, the interesting thing about this is that we see almost the identical effect if we block the synthesis of estrogen. Now, the drug that is very effective in blocking the key enzyme in the synthesis of estrogen is fibrazole. So in women, for example, that have estrogen-dependent malignancies, this could be uterocervical cancer, they're often maintained on aromatase inhibitors. And the aromatase inhibitor, not infrequently, is fibrazole. The amazing thing is that if we block this enzyme in the spinal cord, we get an effect which is exactly the same as blocking the estrogen receptor alpha. So what this tells us is that in this particular situation, estrogen is not coming from the ovary, it's not coming from the periphery, but it is coming from the spinal cord is making its own estrogen. And in fact, I will tell you that aromatase and estrogen receptors are all throughout the spinal cord and they're all throughout the brain. So in a sense, the brain is an organ that can synthesize and make its own estrogens. Now, the other important detail, so in diestrus, it's the spinally produced estrogens that act via spinal estrogen receptor alpha that really clamps the EM2 analgesia. And when you block this, you eliminate that. Now, the other thing I'll point out is that this effect is quick, because within 5 minutes of blocking the estrogen receptor alpha, you get this rise or the unveiling of the EM2 analgesia. Now, why is this important? Why is the quick onset of blocking the estrogen receptor alpha so critical? That's because most of you think, and it used to be dogma, that estrogens act as transcriptional repressors. They basically, or regulators. They turn off or they turn on or they turn off gene expression. And this effect, if it's acting in the nucleus, takes about 18 to 24 hours because you have to have transcription, translation, you have post-translational modification, the protein that is made has to be packaged, sorted, all that takes at least 17 to 24 hours. But it's also known, and this was a revolution in the estrogen receptor field, is that some of these nucleoreceptors leave the cytoplasm, they actually traffic out of the nucleus, through the cytoplasm, and they localize in specific areas of the membrane that are known for quick signaling by a myriad of different kinds of receptors. Whereas this effect on transcription takes 18 to 24 hours, this effect can happen within seconds to minutes. And so what we could say is that since this is happening so quickly, we're not dealing with a nuclear effect, a transcriptional regulatory effect of the estrogen, but we're dealing with membrane estrogen receptor alpha. Now, the other thing is that it's not only the estrogen receptor alpha that's involved, because we discovered that there's also a metabotropic glutamate receptor. Now, the drug that is very specific for this is abbreviated here as YM. And you can see that in the absence of this in diestrus, you get no response. But if you now inject into the intrathecal space this compound, which blocks the metabotropic glutamate receptor, together with EM2, once again, we unveil a response. And the very striking feature about this, the very striking feature is that this is almost the timeline for this effect is essentially superimposable with the timeline for blocking estrogen receptor alpha. And so what this told us is that suppression of EM2 analgesia during diestrus requires mGluR1 as well as MER alpha. And the second thing is that the fact that these timelines were almost essentially superimposable suggested to us that maybe the mGluR1 and the MER alpha were actually physically connected to each other. And the term that is that it could be dimerized with each other. And I'm not going to go into a lot of detail, but I'll just tell you that one of the main biochemical ways to differentiate this is to do what is called a pull-down experiment or a co-immunoprecipitation. So what you do is you solubilize some tissue. In this case, it's the membranes from the spinal cord. You solubilize it, and then you incubate it with an antibody. In this case, it was an antibody against this particular receptor, mGluR1. And then this will precipitate it. And then you use a technique called the Western blot, where you run it in a polyacrylamide gel under pressure. And that basically, if the estrogen receptor alpha was really physically holding hands with the metabotropic glutamate receptor, by immunoprecipitating mGluR1, we should be able to also pull down ER alpha. And then what you do is if you have an antibody specific for estrogen receptor alpha, you can stain the gel with that antibody. And sure enough, what you can see is that the mGluR1 immunoprecipitation didn't just bring down mGluR1, but it brought down two molecular forms of estrogen receptor alpha. And because of that observation, we formulated this model. We said we predict that there's going to be a cell in the spinal cord that's going to contain, it's going to co-express the protein or the enzyme aromatase that is also now going to express our estrogen receptor alpha. And it will also express metabotropic glutamate receptor 1, and will also have the mu opioid receptor. That's basically what we predicted. And then if you notice, I drew each of these touching each other. And that's visually a way to say that we're really dealing with a big heterodimer. This is a macromolecule where all of these things are covalently, or at least they're physically touching each other. Now, indeed, we were successful biochemically in proving that this complex actually existed. But the other question that becomes critical is, it may exist biochemically, but is it really positioned in the cytoplasm, in the spinal cord, so that basically can we show in pain transmitting regions this complex exists, and that not only does it exist, but it receives an input from endomorphin 2, because the idea was that we'd have a cell and that the aromatase acting on estrogen receptor alpha and metabotropic glutamate receptor 1 would modify and change the behavior of the mu opioid receptor so that when EM2 was released onto this, it would not act. The response would be clamped. So the way you look at the anatomical distribution of various components is using a technique called immunohistochemistry. And basically what immunohistochemistry does is that this should, instead of saying primary immunofluorescence, you should say primary antibody. And you can actually tag an antibody with a particular fluorophore, so when it's excited, it will eliminate a color. And that antibody will interact with a specific antigen on a cell, and you'll be able to see it by a certain color. More commonly, we don't label the primary antibody, but what you do is you label a secondary antibody. And so the first thing you do is you incubate like a tissue section with the primary antibody. And then what you do is you come in with the secondary antibody that will recognize the primary. And the stain, the tag, is on the secondary antibody. And by tagging secondary antibodies with the term used as a fluorophore, you could actually see different colors, and the different colors represent the different antigens that are going to be present in the cell. And so remember, what we want to see is that can we find in the spinal cord cells that co-express the mu-opiate receptor, metabotropic glutamate receptor 1, membrane estrogen receptor alpha, and aromatase. And then can we show that such a cell receives an input from a neuron that is expressing endomorphin 2. And indeed, we were able to do it. And basically, in this picture over here, the blue represents the mu-opioid receptor. So you can see this is a cell that is ringed with mu-opiate receptors in the membrane. In this particular image, the green is aromatase, and the red dots are metabotropic glutamate receptor 1. Now, if you take a very thin section through this and stain it so that we know that we're dealing with the same cell, you could say that this same cell also contains estrogen receptor alpha. So here we have a cell that contains a mu-opioid receptor, aromatase, mGluR1, and estrogen receptor alpha. And then we were able to show that this cell actually receives or makes contact with an endomorphin 2 neuron, and that EM2 neuron here is shown in green. And so basically what we were able to establish is that there's, in the spinal cord, there is neuronal co-expression of the various necessary components, such that it's likely that regulation of the responses to spinally injected EM2 likely mirrors that of endogenous EM2. That's what we were trying to establish. It's great to show that we can manipulate responses to exogenous EM2, but the question is, does this really have any relevance to what's regulating endogenous EM2? And the answer is, based upon this anatomy, the answer is yes. So what I've just told you is that basically, if we get rid or we block ER alpha, mGluR1, or we inhibit aromatase, we'll get rid of minimizing EM2 analgesia, and we will unveil spinal EM2 analgesic responsiveness. So these were the predictions. We predicted that if we block this ER alpha, or individually if we block mGluR1, or if we block spinal aromatase, okay, if we block any one of these things, then basically we're going to unmask endogenous spinal EM2 analgesia. And that really is what we wanted to do. Can we tap into an endogenous opioid system and produce analgesia? Because if we could, then in theory, that would take the place of prescription opioids. So the question is then, how do you do it? Now, bear in mind that what I've shown you is unmasking the response to injected EM2. So it's not really releasing. We didn't think that any of these manipulations would release EM2. We just thought that if the EM2 was being released, then if we block ER alpha, or if we block mGluR1, or if we inhibit aromatase, we would magnify the effects of this. Now, one of the basic properties of opioids is that they're not tonically active. So if I injected myself or any one of you with a narcotic antagonist like Narcan, it would have no effect. You would not know the difference. However, if we did something to perturb the system, you would increase the activity of the endogenous opioid system. And of course, that's exactly what happens when patients have pain or they have chronic pain postoperatively. And so there are many different techniques of kind of coming up with a model for chronic pain. The one that we use is basically referred to as spinal nerve ligation. And what is known that is if you cut the spinal nerve, basically, you induce a situation which is called allodynia. Now, allodynia is a situation where a stimuli that is either totally innocuous or it's actually pleasant now becomes aversive. So, for example, if you took a feather and you stroked the back of your hand, either you wouldn't feel anything, or you could say, you know, that feels kind of nice. But in an allodynic state, that stroking of your skin with a feather would be perceived as being painful. It would be aversive. And you can see here in this diagram, the technique for doing this is we don't use feathers, they use von Frey filaments, and the filaments are made up of different stiffnesses. And basically, you know, you apply the filament to a particular part of the body. In this case, it was the hind paw. And it was the hind paw that was ipsilateral. It was on the opposite side to the spinal nerve that was cut. And you could see here that whereas the threshold, you can take a pretty stiff von Frey filament to cause the animal to move its paw, that threshold becomes dramatically lowered after the spinal nerve cut. And so in this scenario, if you lower the threshold, you are making something more allodynic. And if you increase the threshold, then you're making less allodynic. And so basically, we made three predictions. First, this reminds you that in diestrus animal, you know, there's no response to injected EM2. So if there was any endogenous EM2 that was being released, it probably wouldn't be doing you very much good. But if you block estrogen receptor alpha, you now unmask a response. So in our chronic pain model in diestrus, you know, what we were able to find is basically in the presence of MPP, you can see here the threshold was elevated. You compare this to the open circles, which is just a vehicle. And so basically, the animal becomes anti-allodynic or somewhat analgesic. Now, this doesn't mean it's endorphin mediated, but the amazing thing is that you block this elevation by injecting Narcan into the spinal cord. So Narcan is working in the absence of any injected opioid. So we have now unmasked an endogenous opioid anti-allodynic response. If you remember, we saw exactly the same profile with if we inhibited aromatase with fydrazole. And so this is, again, this is what you see in the absence of fydrazole and diestrus. You get no response. If you increase it, you could see, if you now do this in the presence of fydrazole, which is the aromatase inhibitor, you elevate the allodynic threshold. So you will now produce an anti-allodynia. And this anti-allodynia is completely eliminated by the spinal injection of naloxone. So once again, we have produced an opioid analgesia in the absence of any injected opioid. And lastly, you could see, reminds you that we have no response in diestrus, but if we block metabotropic glutamate receptor 1, we get this beautiful response to injected endomorphin 2. And again, you could see that in the absence of any mGluR1 block in diestrus, we get no response. After we block mGluR1, we unmask a nice analgesic response or anti-allodynic response, a membrane estrogen receptor alpha, or by inhibiting spinal aromatase, we can actually unleash, during diestrus, an endogenous opioid antinociception. So the take-home message would be that intrathecal treatments that alter EM2 analgesia also alter spinal nerve ligated induced mechanical allodynia. And this certainly underscores the relevance of manipulating endogenous opioids to pain management. The second thing is that drugs that activate endogenous opioids would enable accessing the pain-relieving effects of narcotics or opioids while limiting opioid addiction and abuse of prescription opioids. The third point is that the imperative to further develop ways to harness endogenous opioids is underscored by the very long-lasting deleterious effects of undermanaged chronic pain. And many of you grew up thinking that with the adage, no pain, no gain. That is wrong. Pain is debilitating. It impedes recovery. It impedes healing. Pain is bad. And by minimizing the use of narcotics in order to alleviate the opioid epidemic is substituting one evil for another. You may be avoiding death by opioid overdose, but now you're undermanaging pain, and harnessing endogenous opioids may be an alternative. And lastly, and I didn't get time to really go into this in detail, but targets for activating endogenous opioids vary with physiological state. And one of the things that we now know is that the targets can vary over the menstrual cycle, or actually the estrous cycle in the rat. And that targets that are effective in one estrous cycle or menstrual stage need not be effective in another. And so that really, really underscores the need, if you're going to go this route, to tap into endogenous opioids, the need for personalized medicine. And lastly, clearly I didn't do the work, but these folks did. They're an extraordinary group of people. Nejiang Liu is a faculty collaborator, and he is singly responsible for all of the behavioral studies that I described to you. Samita Chakrabarty is a biochemist par excellence, another faculty member who I collaborate with, a lot of her contribution showing the formation of these large macromolecular complexes came from her work. Amelia Storman and Arjun Kumar were two MD PhDs in my lab. Arjun is now doing a residency in psychiatry. Amelia went back to medical school. She's now completing her third year. Vijaya Murigan was a postdoc in my lab, and she was seminal in showing the effects of diestrus and proestrus on the macromolecular signaling complex. Alina is a technician in my lab, and Martin Wessendorf at the University of Minnesota and his then technician Steve Snell did all of the beautiful immunohistochemistry. I only showed a very, very small part of what they did. And needless to say, none of this would have been possible without sustained financial support from the National Institute of Drugs of Abuse. And so I thank you very much for your attention. And before I leave for questions, I want to emphasize that if any of you are sufficiently interested, have questions which can't be answered in this time frame, I hope that Carmen will share my contact information with you, and I welcome you to email me, and we'll set up times either via email or Zoom or cell phone, to discuss this further. But thank you very much. I appreciate this opportunity.
Thank you so much. Just thank you for wonderful talk, and I really learned quite a lot about the interactions between estrogen and the opiate system, which I was not familiar with. Now, I have one question that was raised in the chat, Dr. Glitzer, and it says, when you spoke of the data obtained from pregnant women versus non-pregnant women at the beginning of your talk, it reminded me of the role of oxytocin in the process. So as you know, oxytocin is analgesic and it enhances the mu receptor signaling. I'm hoping you could talk about how you see the role of oxytocin and if it could help to amplify the endogenous opioid response.
Thank you. That's a great question. To be honest with you, most of the work that I did during gestation and the hormonal simulation of gestation really led to the days leading up to actual parturition. And I believe that the work that was done in the pregnant women by Kogan and Spinato some years later also looked at the last trimester leading up to, but didn't include actual parturition itself. I do believe, or I'm trying to now remember, that basically there are additional dimensions of this antinociception that builds onto what I described and what Kogan and Spinato described. And if I'm remembering correctly, this actually coincides with the actual periodic contractions of the uterus, leading to actual, during labor and delivery. So I would actually think that the involvement of oxytocin would probably be superimposed on the developing of the antinociception, during the third trimester.
Okay.
But, so that's a great question.
Yes, I am also very interested in the role of oxytocin, but more at the central nervous system, in particularly in the nucleus accumbens. But there's another question. It says, which I thought is also very pertinent, what would you predict in a non-cycling female? For example, an over-optimized or a menopausal woman. Would they be similar to pregnant subjects, or would a lack of estrogen change the receptor interactions?
Another great question.
Mm-hmm.
And I've thought of it, so much to do and so little time. We have not looked at postmenopausal rats, okay, but clearly one should. I think that the whole arena of menopause and pain, knowing how integral and involved estrogens are in the brain in actually modulating nociception, one would think that basically it is definitely a critical area to look at, because basically I think it's assumed that in postmenopausal women, the pain sensitivity would be basically extrapolated from what you see in regular cycling females, and there's no reason to suspect that, no reason whatsoever. So we have not done those experiments, and I think that there's very, very little in the literature. What I would point out is one of the complications that I think really has to be considered is that think of women that have estrogen-dependent malignancies. Invariably, they're maintained on aromatase inhibitors, and these aromatase inhibitors do indeed penetrate the blood-brain barrier. And so when you're inhibiting ovarian aromatase, you are also inhibiting aromatase in the spinal cord and then in the brain. And that really behooves somebody to develop a peripherally restricted aromatase inhibitor that will act peripherally, in the ovaries, but will not penetrate the blood-brain barrier. And so one can imagine, and I don't really know the literature on this, but I just wonder if women treated with aromatase, if studies have been done looking at pain sensitivity in women maintained on aromatase inhibitors. I think that really should be a study that should be done.
Okay, and there's another question. Is it possible to engage the placebo effect as an analgesic?
Well, the answer is yes, because certainly those early studies have done that. They've shown the effectiveness in actually maintaining, behavioral analgesia, like in the placebo dental pain studies, and they've also shown it by imaging opiate receptors in the brain and showing that the release of endogenous opioids. The problem is in the reliability and in the predictability of doing that. So that's the question. I mean, if you're post-operative and you really have severe pain, do you really want to go the route of trying to elicit placebo, analgesia, which I would imagine would be very, very variable and not always predictable. I think a better route would be to try to map out the various components that lead to placebo analgesia, and then to figure out ways. So with a drug, with a pill, can you target that? Now, the interesting thing is that I showed you the analgesic effects during a particular estrous cycle stage of fydrazole. Well, fydrazole is FDA approved because it's currently used. It's used to treat ovarian and uterocervical cancer. And so one certainly might think of, injecting fydrazole in the spinal cord, okay? And there are other drugs like estrogen receptor alpha antagonists. Also, I think some of them are FDA approved. And so basically, I think it would be very worthwhile to do a study where you test the analgesic effectiveness of these compounds, which should not have a direct analgesic effect, but should act indirectly by amplifying or magnifying the endogenous opioid analgesia.
Okay, and then we have one last question. It says, is there a correlation of symptoms of myofibromyalgia and the women reproductive cycle?
I don't know the answer to that. I don't know the answer. I would strongly suspect that basically that study has not been done. Although, one could easily, when I'm finished, I'm going to actually go do a PubMed search on fibromyalgia, okay, and a stage of menstrual cycle. I think to see, because I don't know the answer to that, but I suspect that by and large, a lot of pain research has not really attempted to correlate stage of cycle with severity of pain symptoms. Although I will tell you that basically, in terms of the chronic pain literature, I think to the extent that women experience this, it's really the most severe around menses. And that's the time in the menstrual cycle that I believe is most similar to diestrus in a rat. Okay. It would be very useful to do that study.
Okay, thank you. So I think we will finish here because we're already at 6 o'clock and we promised to finish on time. We will convey to you the other couple of other questions that we have for you, Dr. Ginsler.
Sure.
We really appreciate you taking the time to being with us today. From here, the NeuroID program at the University of Puerto Rico, Rio Piedras, we thank you so much to being with us, and please join us for the rest of the series that we'll be having every Wednesday at 5 through this platform. And next time we'll have our very own Dr. Jose Garcia Raraz to give his presentation and talk about neurodegeneration. So once again, thank you so much for all our colleagues and collaborators, and of course the BP Endure program for supporting us in this venture of a virtual summer. And
well, truly my pleasure, truly my pleasure. And make sure that I'm on the mailing list so I get the announcements of the upcoming seminars.
We'll make sure you will be part of it. Thank you so much for being with us.
Okay, my pleasure. Bye-bye.
Okay, take care all. We'll see you.